Hello, my name is M.Grande, and I thank you for coming to listen to my presentation on the physiological understanding of human muscular disrepair and recovery. But firstly, for every 30,000 people, one of them will have some form of muscular dystrophy, which is a fatal disease that slowly murders its victims by disintegrating the muscles. Now, muscle lesions, on the other hand, while being less horrifying, can comprise from 10% all the way up to 55% of all injuries people experience, as well as being extraordinarily expensive, slow, and gruesome to heal. Taking a deeper dive into the former, muscular dystrophies, such as the most prevalent, Duchenne's muscular dystrophy, are genetic impairments that are usually fatal before the age of 30. Attacking the dystrophin protein, Duchenne's muscular dystrophy weakens the muscles until the subject is effectively immobile, paralyzed, and then killed once the disease reaches the lungs and heart. The trickiest thing about treating DMD is how large the dystrophin gene really is, making it difficult for gene therapy to tackle the problem. Moving on to muscle lesions, muscles are one of the easiest parts of the body to tear or break. Of said injuries, natural healing takes a ridiculous amount of time and money, not to mention that most injuries to the muscles very rarely heal completely due to scar tissue. What's tricky about muscle injuries is how prevalent they are in those who play sports, making it almost impossible for a professional athlete to go a season without one. My first article has to do with this issue, titled New Strategy to Facilitate Muscle Regeneration After Injury, brought to you by the University of Barcelona in Spain, with some notable authors being Teresa Pages and Joanne Torella. Some notable takeaways from this article are that hypobaric hypoxia conditions have been proven to have a beneficial regenerative effect on the body when cells are forced to adapt to a lower oxygen intake, which is exactly what hypobaric hypoxia conditions are. Now for the why, it's that the HIF protein in cells activates metabolic changes when exposed to an oxygen deficiency, such as activating an endothelial growth factor, which can also activate other growth factors, contributing to healing. The HIF protein also activates many enzymes, mitochondrial efficiency, and protein synthesis, which all contribute to muscle regeneration. Moving on to my second article about muscular dystrophy, new gene therapy for muscular dystrophy offers hope, brought to you by the Washington School of Medicine in the United States of America, with a notable author being Dr. Haikem Tassifau. Some notable takeaways from this article are that gene therapy utilizing small gene packets and shuttle vectors to replace a defective DMD gene are being studied. Now, if you remember earlier, I said that the trickiest part about treating Duchenne's muscular dystrophy is how large the dystrophin gene really is, but the scientists in this article tackled that problem by using 3 harmless virus shuttle vectors, each with a third of the dystrophin gene, so that it can be reassembled inside the body. Improvements are still being undergone, such as lowering the number of shuttles needed so that the immune responses are a reduced risk, but the results are promising. As a third article, it's very similar, using gene therapy, but this time with cardiac muscle. It's titled Promising New Gene Therapy Approach for Genetic Heart Disease, Clinical Trials Imminent, brought to you by the Hubrecht Institute in the Netherlands, with a notable author being the one listed on screen. Some notable takeaways from this article are that proven in a laboratory environment on mice test subjects is a new gene therapy for the genetic disease ACM, which has successfully restored protein levels and rhythmic contractions. For those of you wondering what ACM is, it is a cardiac muscle disease of the heart that poses a risk for sudden cardiac arrest, and seen in the picture, arrhythmias, which is an irregular heartbeat. And now, unlike DMD, this process works by using a similar gene which codes for a similarly functioning protein, while DMD used the exact gene produced in humans. Next steps outlined by the article include investigating clinical potential, safety concerns, and efficacy on human patients. From these articles, some research questions that I now have are the following. Number 1, would protein injections, gene therapy, or, recently learned by me, CRISPR gene editing work best to treat muscular diseases? 2, how could I do a research project about muscular injury recovery using hypothesized methods while at the same time meeting New York State humanitarian animal laws? And 3, how much of a difference is it to treat skeletal muscle injuries compared to cardiac muscle injuries? And what about genetic diseases? For those of you who want additional resources, they are on screen. And with that, I conclude my presentation. Again, thank you for listening.